Yeah, okay. So yeah, let's get started on lecture 3, which is a continuation of sort of the imaging and calibration process for interferometers. And then switching at the end, just a little bit of content on sort of radio emission mechanisms and sort of their observational properties in terms of radio astronomy. Right, so yeah. So yeah, in today, I wanted to slip in one or maybe 2 slides on correlators quickly, because I realized I'd wanted to mention that last week and forgot. So just a little bit on that. And then back into imaging, weights, deconvolution. I took out the bits on sort of sensitivity and noise in interferometers, because that's, I think, covered reasonably well. Oh, sorry, I'm getting background noise. Who's that? Adese, could you mute? Thank you. Okay, yeah. So I've taken out the bit on sort of sensitivity and noise, because that's covered, I think, reasonably well in other places, and I didn't have time for a discussion on sort of missing scales in interferometric images, although we did discuss that a little bit last week. So yeah, put in a section on calibration and radio emission mechanisms. And I did mention this in the Slack, but today is the deadline for deciding credit versus audit for the course. I'm going to send the list of crediting students to Margaret probably tomorrow. So I've basically taken what you said when you signed up for the course before. So if you want to change that, today is the day to do it. Yeah. Okay. So yeah, correlators, which are sort of the devices required for sort of the cross-correlation aspect of the interferometer. So the process of measuring visibilities means we're correlating signals for any given baseline from any pair of antennas. And so this means that if we have n antennas, we get this n minus 1 over 2 separate baselines. And assuming that we have sort of two orthogonal antennas to get the full polarization, you end up with 4 correlations on each baseline. Like if you have a X and a Y dipole antennas, then you correlate the X of antenna A with antenna 2, or the X of antenna 2, the Y of antenna 1 with the Y of antenna 2, but you also correlate the X and the Ys from the antennas as well. So you get the 4 correlation products to get the full Stokes information, the full polarization. And so this ends up being a lot of computations, essentially, in order to do this in real time. And so a correlator effectively is a small supercomputer, or not even small in some cases. In some cases, it's a proper supercomputer to manage all of the data that's coming in, to manage all of the necessary correlations, and sort of other computational steps that are required. And so there's sort of two ways that these calculations can be structured, which gives you 2 types of correlators that people sometimes talk about. So in the previous lecture, I talked about taking a time series and correlating them by sort of multiplying the 2 voltage signals together and averaging. But the alternative to that is if you Fourier transform your signal, which we do when we're making channels during the channelization process, you've transitioned then from sort of the time domain into frequency domain, and the convolution becomes a product in the corresponding Fourier domain through the convolution theorem. And so instead of convolving the 2 signals, you calculate the Fourier transforms of each one separately and then multiply the Fourier transforms together to get the visibility out. So you get these 2 types of correlators, the XF or FX, where the X is the cross correlation and the F is the frequency, the channelization FFT. And so you can correlate first. You can correlate the signals with each other or the autocorrelation for every baseline, and then take the FFT of that to get the frequency structure out. Or in an XF correlator, you Fourier transform everything, and then you take advantage of this convolution theorem and just multiply the Fourier transforms, as I described. And so the output of both types is that you get visibilities out. And so it's just two different ways of calculating the same thing, and it doesn't change things much on the astrophysical side of things. But sometimes you'll see people talking about what kinds of correlators they're using for different telescopes and such. So if you see the terminology, it's not really so relevant astrophysically, but now you have some context. So let's go back to the discussion that we ended on last week on imaging. So I mentioned, I had this slide last week, but just a quick refresher. So the imaging process involves, we want to make a image with some number of pixels. And we get this by applying an inverse Fourier transform to the visibility data that we have. And so the user then sets how many pixels and how large they want each pixel to be. And then that then sets how much of the sky is covered and sort of how well sampled the image is. The FFT algorithm is then usually used to do this inverse Fourier transform. It's the most efficient. And to get this n by m pixel image, you require an n by m grid of points in the UV plane. And so there's this step of taking the visibility data and sort of dropping it onto this fixed grid of positions in the UV plane, this gridding process. And so as sort of part of this process, or as part of the inverse Fourier transform, we can apply this weighting scheme. So I mentioned this before in the context of just having finite baselines and how this gives you your synthesized beam from the sort of lack of information that you get from missing baselines. And so, yeah, so we defined this previously as you have a measurement for everywhere in the UV plane that you have a baseline between telescopes ij, you have a baseline with some UV coordinates, and so you have a measurement at that location in the UV plane. And the ensemble of these is your weighting or sampling function. But we do have the freedom here of assigning a weight to every visibility. Or this can be done after the gridding step. You can apply it on the visibility grid. And the effect that this has is it modifies the shape of the synthesized beam. Is you can choose to sort of upweight and downweight sort of different baseline lengths or different parts of the UV plane. And this then changes the shape of the synthesized beam, and you can sort of choose certain parameters that you would like to optimize for. Things like, do you want to optimize to have the lowest noise in the image, for example, versus the sharpest possible image with the available baselines, things like this. And so these weighting schemes are automatically implemented in most modern imaging packages, where you can sort of select from a list of implemented weighting schemes, and it sort of automatically calculates what the weights should be for your data according to some scheme. And so some of the more standard weighting schemes I wanted to discuss, starting with the natural weighting scheme, which is the noise minimization scheme, essentially. And so that follows kind of a, I want to say least squares kind of optimization, is that true? I think that's true. Where you basically weight by the inverse variance. You have some estimation of the noise, the variance on every visibility or every grid point, and you just downweight the really noisy points, and you upweight the points that have really low noise. And the net effect of that is it minimizes the the noise in the resulting image. And one of the tendencies that this tends to have is that it usually ends up downweighting the long baselines, because you tend to have, for most array configurations at least, you have more short baselines than long baselines. And so you then have sort of more information on the short baseline sort of scales. And so the long baselines tend to get downweighted slightly, with the effect of reducing your resolution. So you get the best noise possible for the data, but you lose some of the resolution. And sort of the sort of opposite of that then is the uniform distribution, where you just weight everything equally without accounting for sort of the density or the comparative noise on the different measurements. It's sort of, this is kind of the simplest weighting scheme, where it doesn't actually apply any weights for taking into account any additional information. So everything is sort of treated equally, and so the long baselines get equal treatment to the short ones. And so you do get you prevent this sort of loss of resolution, but you're also not downweighting noisy measurements. And so you potentially pay for that in the image. So between these two, there's something called robust weighting, and there's a couple of different forms of robust weighting. The one in CASA, at least, is Briggs weighting. And robust weightings are basically using some kind of an equation that has a free parameter that you can use to adjust somewhere between natural and uniform. So you get this sort of robustness parameter that the user selects, and depending on the value, it'll weight things in a way that looks either a little bit closer to uniform or a little bit closer to natural, or somewhere in between. And exactly what values you should use under what conditions and sort of how strongly they affect things depends a little bit on the implementation, and it's it's a little bit arbitrary sometimes, and so there's not always it's not always clear what the best value is. So there's some amount of trial and error, and some amount of sort of user experience goes into selecting a robustness parameter. But the net effect is you tend to get sort of some of the advantages of both. You can sort of reduce the noise while keeping the resolution okay. And sort of the last of the common sort of weighting schemes is the Gaussian taper. And this is one that's actually applied on top of the other schemes. So you can apply one of the first three and then add a Gaussian taper on top of that. And what this does is it deliberately downweights the longer baselines. It basically takes your UV plane and applies a Gaussian across it in terms of the weighting scheme, so that all the things that are far away from the origin, all the long baselines, get reduced in their weights. They're considered sort of less, they contribute less to the final result. And this deliberately reduces the resolution of your final image, but it's also a very good method to reduce the side lobes in the synthesized beam. And it does have some advantages in terms of reducing noise on small scales. And so a Gaussian taper tends to be used most often when you want to deliberately reduce the resolution in order to maximize the ability to see things like extended sources. Like if you think you have a source that's kind of large and fluffy compared to your synthesized beam, you can apply a Gaussian taper and it gets rid of some of the side lobe problems from the beam and helps sort of bring out fainter sources a little bit better. So I've stolen some examples on this. This is from a slide taken from David Wilner from the NRIO Interferometry Summer School from a couple of years back. And this kind of demonstrates the same data imaged with sort of different weighting schemes. And so on the top left is sort of the natural weighting scheme, where the resolution, this is the beam size, the major and minor axis, is largest, but the noise, the RMS in the image, is the best that you can achieve. Contrast that with the uniform, which gets you a much smaller beam size. It's almost half the size, so your resolution is improved by almost a factor of 2, but so is your noise. And so that's a trade-off. And which one of these you might want to use depends on your scientific goals. The robust, so for robust equals 0, which is kind of in between these two, you get most of the improved resolution from the uniform, and you get most of the improved noise from the natural, but it's not as good at either as a sort of a more pure weighting scheme. And if you take the robust weighting and apply a taper to it, they've tapered it deliberately down to the same resolution as the natural image. The RMS isn't as good, but you can see in the image that the side lobes are sort of better controlled, and so you get sort of a better sense of the actual sort of what the source looks like, and it sort of beats down the structure and the noise a little bit more. Okay, any questions on that? Okay. So a sort of variation on regular imaging that I wanted to mention is multi-frequency synthesis, which is kind of an improved technique. And so the idea behind this is that for every baseline, if you are making measurements over some reasonably broad bandwidth with many channels, every channel, every frequency ends up having a slightly different length in the UV plane. Because remember that the UV coordinate system is the physical baseline length divided by the wavelength. And so you change the wavelength, you change the UV coordinates, because the physical length of the baseline doesn't change. You know, the telescope is fixed. And so for every channel, it's sampling a slightly different point in the UV plane. And so when we want to go and try and fill in the UV plane to maximize the information we have for imaging, that's potentially an extra source of information, except that most of the emission will have some kind of frequency dependence. It'll have a spectrum. And so that means that you can't just take visibilities at different frequencies and sort of combine them together, because at some frequencies you might have a signal that's bright, and the other end of the band, it might actually be weak just because of the change in the spectrum. And so there is a trick that sort of gets around this, and this is the multi-frequency synthesis method, which assumes some kind of model for the spectrum, for the shape of the spectrum, with some model parameters. And it can go through the imaging process where the output is not sort of directly the intensity, but it outputs images of the model parameters. And so the typical form for this then is you assume that most of the emission in an image is going to follow some kind of power law with some intensity at the reference frequency and some spectral index. And both of these change as a function of which pixel you are in the image. So different sources can have different spectral indices and different brightnesses. And so somehow, and I confess I don't really understand the mathematics of it, somehow you can produce images of those directly. And so you're taking advantage of the full information across the entire observing band to produce a single image that captures all of the spatial information from the different frequencies, as well as all of the spectrum information across the entire band together. And so the typical form of this is sort of intensity and spectral index, but in principle, the software allows for an expansion to have additional terms. They often call these Taylor terms because they do this Taylor expansion in the logarithm, where they take this expansion where you have the 0th order term is just your intensity, 1st order term is your spectral index, following from the first equation. And then you can just tack on extra terms that depend on like logarithm squared, logarithm cubed, etc. So this would be like a spectral curvature parameter. And so when you have the first two parameters, you're assuming that your spectrum is a straight line in a log-log plot. If you add this, you can sort of model quadratics in a log-log plot. And in principle, you can go to higher orders, but they tend to get very noisy and unreliable. And so most people will image these first two parameters, or sometimes they'll even drop spectral index if they think it's not interesting or important. Yeah, and so it should be noted though that this is only really useful for continuum radiation, so things like synchrotron or thermal emission. It doesn't work for spectral lines, for example, because those are by definition very narrow in frequency. And it doesn't work for polarization because polarized signals don't follow power laws. You have things like Faraday rotation that mess up things, so you have a much more complicated spectrum. Okay. Any questions on that? Okay. So sort of a slightly tangential point I wanted to make, because this is something that was pointed out to me by one of my professors years ago and isn't really talked about a lot. Which I think I talked a little bit about this last week, which is that integrated fluxes of sources are surprisingly hard to measure for various reasons. One of the interesting problems of trying to measure, like if you have some kind of like a resolved source, like maybe a nearby galaxy or something, just trying to measure how much radio flux density, I should say, flux density. So you're integrating over the parent size of the source. And that turns out to be surprisingly tricky. And one of the reasons for this is that an uncleaned image, and we're getting into the clean algorithm soon, so just an image that comes out of one of these imaging algorithms always contains zero net flux density. If you integrate over the entire image, you always get zero signal. Can anyone guess why?
Does it have to do with the side lobes?
Not directly. Well, it depends what you mean by side lobes, I guess. Yeah, I guess it's fair to say it's related to side lobes of the synthesized beam, yes. I guess. Anyone else have thoughts? Putting you on the spot, maybe that's a little unfair, but it's a weird result, but it's not actually. It sort of comes out of the mathematics fairly straightforwardly. So if you think about the imaging process, it's a Fourier transform or an inverse Fourier transform. We're building a signal on the sky out of sine waves, right? For every location in the visibilities, because it's a Fourier transform, you're building up sine waves into some combined signal. And every one of the sine waves that you're putting in when you reconstruct the image is, by definition, it's a sine wave, so it has zero mean when you sort of integrate over the full, however many cycles of the sine wave. The only reason that you sort of see sources in different places is because the phases of the Fourier components line up such that at some places everything interferes constructively and you get a big positive peak, at the location of a source. But that is just then balanced out off the source. You get all the ripples, the side lobes, and they have a lot of negative components, which are unphysical. And so in any interferometer image, normally you'll get all these regions of negative intensity, which are indications of sort of properties of the measurement and not any sort of physical meaning. And this is because normally if you did a Fourier transform of some signal or some image, whatever mean signal is present across the entire image, Fourier transforms into what would be the zero frequency Fourier component, normally, which is the origin in the UV plane. So U equals 0, V equals 0, which is a baseline of length 0, which we don't actually measure normally. Because the idea of a baseline of length 0 is a little problematic in some ways. And so what we're basically losing there is we're losing the information on sort of any mean signal across the sky. That information is gone. And so when we do the reconstruction with the information we have, we've lost information on some sort of like 0 level offset from the mean that should be present in the image, and all we're left with is sort of the fluctuations about the mean. And so this kind of comes back to sort of the missing scales problem of not having this sort of 0 length measurement. And so your synthesized beam, as a result, if you integrate over your entire image, it goes to 0, and every source, which is convolved with the synthesized beam, also integrates to 0. But sort of in anticipation of clean, when you do clean and you have a clean beam, that doesn't have a 0 integral. And so what this looks like in an image, and this is a LOFAR image from my PhD. If you have a large object, this is a nearby radio galaxy in the center here. And so a large object, you see a lot of flux, and then off of the object, you then have all of this sort of negative sort of counterbalance to balance out the image. And so you get this, what they call a negative bowl, where if you were to plot a profile of it, is you'd see these big dips to negative and then a big positive feature for the actual source. And so you can imagine trying to measure the sort of integrated flux if you just sort of like draw a circle around it and add up all the pixels. Suddenly it's very dependent on where you put that circle. If your circle starts to extend into the negative bowl, you're adding negative pixels into your integral. And so it becomes a really tricky kind of thing to measure accurately. And so the solution to this, so far as I'm aware, is generally to try and come up with a good deconvolution algorithm that kind of gets rid of this negative bowl. So that you don't have that problem, so that all the real emission is properly positive definite, and so you're not sensitive then to this kind of problem. So it's a bit of a weird effect, and something that you kind of have to be aware of if you just want to try and pull a flux density measurement of a source, particularly if it's a large source. Any questions on that? It's a bit weird.
So just a small question. So if you were to look at the peak of the flux density profile of this source and compare that to the cleaned image, does that mean that it would increase the signal? Like if you would actually have a higher flux density at the peak?
At the peak? So at the peak, probably not. It should be the same if everything goes sort of correctly. I think in general, I mean, I can think of cases where that wouldn't always be true.
Okay.
And that would be things like if you have an extended source and the peak is actually sitting in the negative side lobe of other emission from around it, because the synthesized beam will have some negative side lobe contributions. And so you can think of your peak if sitting, there's other emission sitting just off to the side where that negative bit is sitting on top of the peak and diminishing it.
Yeah, I guess I was just thinking of the simplest of cases, something similar to what you're showing right now.
Yeah, so
I'm just trying to like visualize what's happening in my head.
Yeah, it's a bizarre phenomenon, really. And
Okay.
It's not really that intuitive.
Okay. That's good to know then.
Yeah, no, well, it's interferometry. Nothing is intuitive. But it's one of those weird things that you sort of see it and when you look at it, you're like, okay, I can kind of make sense of why this happens, but how to solve it is not always obvious either. And you can kind of see this, like if you take an image, for example, you take an image that has no cleaning on it, and you look at the image statistics in whatever package you're using, you can just like pick a region of sky, add up all the pixels, and they will add to 0. And that's, that was something that, like when I was a student, I did that a couple of times, and it's like, okay, is this a meaningful result? And I had to sort of stop and think about this in order to understand it. that was a sign that that wasn't actually things going wrong. That was just the mathematics of it. Okay. So yeah, so that kind of launches us into deconvolution algorithms. And so basically what we can say, I mean, particularly based on that last result, is that the convolution between the actual sky intensity distribution, so the actual image, and the synthesized beam, which comes from our measurement, is kind of a pain because it's, making it hard to get good measurements of certain things in the images. And I mean, granted, it actually does represent missing information. That's why we have a synthesized beam, is we're missing information from baselines we don't have. But we would try to improve it in some ways because it's bad, and particularly it's bad for dynamic range, which is the ability to differentiate between sort of faint, actually measure a faint thing next to a really bright thing. And so you can imagine having a super bright source in an image, and so it's convolved with the beam. You can think back to that sort of hex shape, that 6-spoke pattern for the VLA that I showed last lecture. So imagine having a bright source with these 6 spokes coming off. Beside any of those spokes, if there was a faint source, would you be able to tell it was there? Would you be able to reliably measure it and differentiate it from all the artifacts that are the side lobes on these spokes? You generally couldn't. And that's a huge problem. So you're basically losing all the faint emission around your bright sources. And so we want to recover that as much as possible, which brings us to, basically the class of deconvolution algorithms which are trying to remove the effects of the convolution of the synthesized beam. And so this this of necessity involves some kind of assumptions, basically assumptions to fill in some of the missing information that we don't have. And so these can be things like smoothness assumptions, or assumptions about typical shapes of sources, like maybe most sources are unresolved, for example, things like that. So you get different kinds of algorithms. And so every algorithm has its own set of assumptions and its own sort of methodology to try to accomplish this. And there's a ton of them. I actually have no idea how many different image deconvolution algorithms have been developed. I've seen a lot of them. And the biggest complaint, this is a pet peeve of mine, is that people will write papers saying, hey, I have applied my favorite image analysis, information theory algorithm. I've applied whatever my favorite algorithm is to deconvolution of radio images. I've, developed my own little software package to do this, and I've tested it, and here's how it does way better than other algorithms. And then they never give the algorithm out. Like they publish a paper saying, here's the algorithm, I wrote a code, it works, but they never share their code. And so they never develop a tool to let other people use their algorithm on their own data. Or if they do release a tool, it's usually not very user-friendly, or it doesn't support most data. And so there's all this work in sort of algorithm development, and none of it ever goes anywhere because no one does the implementation. And so that's a pet peeve of mine. And so as a result, the only really common algorithm in use today is clean and its variations. And that's because clean is built into most of the imaging software that gets used. So CASA and other imagers basically all have clean, because that was sort of one of the first ones developed. And whenever a new one comes out, it's always compared to clean, and usually it does better, but it's never, people don't build these into CASA, so no one ever gets to use them. So all we're really going to talk about today is clean. That's all we really have access to. So clean, and in particular, starting with the traditional clean, which is the Hoogbom clean. I don't know if I'm pronouncing that right, but this is from 1974. And so Hoogbom clean is a iterative algorithm, and the key assumption is that the sky is made of a collection of unresolved sources. Basically, it tries to model the sky as a collection of little delta function sources at different locations, and sort of replace all the sources that are convolved with the synthesized beam and replace them with effectively delta functions, or something that sort of at least has a more desirable beam shape. And so I'll sort of walk through the steps in that on the next slide. But the basic algorithm works purely in the image plane. Is you just work with the image and then try to improve it. There are some modern variations that actually work on the visibilities, and they tend to give some better performance in some ways, and so they tend to be more common. But I also, one of the tricky things with clean is there's a lot of free parameters and a lot of sort of variations on the technique. And so there's free parameters like how many times you iterate through the algorithm. There's a gain factor in the algorithm. There's thresholds for how long you run clean for and how much flux it's allowed to process. There's things like clean boxes, multi-scale clean, variations on the technique. And I'm going to talk a little bit about the more common ones, but there's a lot to it, and using it in the best way is a bit of a black art. And it's kind of one of those things that people just pick up some experience with as you go. And so that's kind of one of the frustrating aspects of radio astronomy is no one can tell you what the best way to clean your data is, sort of a priori. It's something that is somewhat unique to every data set, how to do it best. And so there are pipelines to do it good enough, but if you're processing your own data, sometimes if you want to get the best possible image out, you kind of need to do it by hand and tune and tweak things and experiment a bit. And that's sort of the traditional way of doing data processing with interferometry data. All right, so let's go through sort of the clean loop, the iterative loop. So you start with a residual image, which is initially just your initial dirty image, so the one that you get out of the imaging process. And you create a new blank image which holds your clean components. And so from there you go through sort of an iterative process where you find the brightest point in the image, and you assume that's a source, a delta function source. And so you basically say, okay, if this is a delta function source, then you convolve it with the synthesized beam and say, okay, this is the side lobe pattern I should be getting from that source. And you say, okay, I am going to remove that side lobe pattern. And so you subtract it off by some fraction. So there's this gain factor, which is how much of the source you want to subtract off in one go. And so in principle, you could subtract 100% of the source flux in one go and get rid of it in one iteration, but this can be a little unreliable if there are secondary effects, like if your source is sitting in a side lobe of a neighboring source, that can change what its actual flux looks like. Or there's other kinds of sort of errors. And so typically you do it conservative, you subtract a little bit of a source at a time, something like 10%. Some people will go a little higher, 20, 30, 50% of a source in one go, but 10% is the default these days. So you subtract off 10% of the source in terms of its full side lobe pattern. And then you record that with a clean component, which is in your clean component map, you put a delta function at that location with equal to the amount of intensity that you've subtracted off. And so that's sort of a record of what you've subtracted off then. Is I subtracted off this much flux density or intensity at this location. And so you then take your subtracted image, which is your new residual, and you look for the next brightest thing. And you subtract off a fraction of that, and you do it again. You find the next brightest thing, and you keep going and going and going and going until either you've reached a cap on how many times you're allowed to iterate, or there's no more pixels that are bright enough to be above some threshold. And so I'll discuss sort of why these are the two stopping criterion momentarily. But you basically repeat until it's decided it's done. And after that, you then create a restored image where you take your clean component model, which is all just delta functions, and you then convolve it with a clean beam, which is usually a Gaussian, and you usually choose it to be a Gaussian that has the same size as the main lobe of your synthesized beam. And then you add that back to your residual image. And so the reason you do that is the Gaussian clean beam is nice because it has no side lobes. It's just a nice smooth function with no sort of weird behavior at the edges. But it does sort of give you sources back that have the same size as your original synthesized beam. And the reason that's kind of important, there's a couple of different reasons. One of them is that intensity scales get a little bit weird because intensity is always in, well, usually at least, in Janskis per beam. And so that's defined relative to some size of beam. And so if you have a combination of emission that's on the size of your synthesized beam, and then you have some emission that's in delta functions, you can't really add those two things together in a meaningful way on a normal intensity scale. And so that makes things a little bit messy. And there may actually be a lot of signal that is not in the clean components. So for example, particularly if you have like a resolved extended source, per pixel, it's really faint. And so it might be below your clean threshold. But you could have a lot of pixels that have a moderate amount of signal. And so it might actually add up to a lot of flux density over all those pixels. And so you can't just ignore the residuals that are left after clean is finished because there's still signal potentially in there. And as well, the clean components may not always be perfectly accurate either, as they can have some amount of position uncertainty or error caused by different things. And so if you created an image where you just left the delta functions in, you might say, oh, this source is exactly on this pixel, but you've lost your kind of margin of error in there. It's kind of misleading. And so this sort of convolution with a Gaussian kind of solves all of these problems. And it makes things more consistent also in terms of how the noise behaves, because the noise, I forgot to put in a slide on this, but the noise is correlated from pixel to pixel. And that plays an important role in some types of analysis of errors, like what is your error on your flux, for example. And so accounting for that properly is better if all the signal also has the same kind of correlation structure, which comes from having a synthesized or a clean beam that are kind of similar like this. Okay. So I have an example of what this kind of looks like in practice, again, stolen from the VLA summer school. And so in the left plot here is a dirty image. So this is the image that comes straight out of the inverse Fourier transform. And so at the start of the clean algorithm, you have the residual map is equal to your initial starting map, and you have no clean components in the your clean image. So you start it running, it finds the brightest pixels, generates clean components, subtracts off the beam. And so here it's put in a 30 clean components, some on the second source and a bunch sort of spread around the center source here. And it's a little hard to tell, but it's kind of cleaned out the flux in the center of this big source here. It's sort of captured all the bright flux. So initially, the color scale is such that this peaks at about 0.1 Janskis. And after 30 iterations, this is now peaking at 0.04 Janskis. So it's removed 60% of the peak flux in the first couple clean components. And sort of, yeah, gotten the brightest parts of the source. Now it runs a bit longer, so up to 100 clean components, and it's kind of picked up a lot around this larger source now. There's a lot more sort of structure around it. The residuals have dropped to 0.01, so they're now sort of a tenth of the original peak. And you're starting to see the noise we're starting to get closer to the noise level here. After 300 clean components, there's basically no structure left in the residual. It's basically almost pure noise now. And by about 500 odd, this is completely noise-like. There's no signal left here, and you can even see there's some clean, it's probably hard to see, but there's some clean components in random locations where it's actually started cleaning noise peaks. Just because if the brightest thing left is just noise, it'll pick that and put a clean component on it and subtract that off, even though it's not an actual signal. And so at this point, the clean algorithm was stopped, and the restored image was created by taking the clean components, convolving them with this Gaussian, and adding on the residual noise. And so the final image looks like this, and you can see it's a lot cleaner than your initial dirty image. It's gotten rid of all of this side lobe structure here. A lot of the messiness around the large source is gone. And so you're left with a much easier image to analyze. Is sort of the end result. Any questions on that, on the clean algorithm?
Do you actually have control over these parameters when you are imaging this?
Yeah. Yeah. So some of the parameters you have to set right at the start. Things like so if I just go off this slide, so stuff like pixel size and visibility weighting, gridding, those are all in the imaging. And so I mean, a lot of, I think CASA does the imaging and the cleaning all in one step, I think, these days. But originally you could do these separately. And then when you get to the deconvolution, things like the iterations, the threshold at which you stop, those are all set at the start of the algorithm. And then you would just have to let it run, look at the result, and then decide if it worked well enough and whether you want to change the parameters. These days, CASA at least has an interactive clean where you can run clean for a couple of iterations, and then it'll stop and it'll show you what it has. And you can decide, is it working well? Do I keep going? Do I stop? Do I go back and change things? You can sort of decide and then keep running it or start again. So there's that control in the middle of the process. Yeah.
All right. Thanks.
Yeah, okay. So there are a number of sort of different complications that can arise during clean. One of which is that setting the parameters can be tricky. It's hard to know what good parameters are sometimes. So things like the number of iterations, it really depends on how big your pixel is and how much signal is present, because the more sources you have and the brighter they are compared to the noise, the more iterations you're going to need to get rid of all the signal. And that's hard to know. But ultimately, the number of iterations only actually matters as a way of stopping the algorithm from running for infinite time. The purpose of the iteration limit is to just stop it from running for like 3 days straight. So you can kind of tune it to decide how long am I willing to let this run? Is the answer an hour? Is it a day? Something like that. And then you have to kind of guess from trial and error how many iterations equals 1 hour or 1 day. And that's yeah, trial and error, because it depends on your computer how quickly it runs. And so that's basically all that parameter is important for. Clean thresholds are much more important and also tricky, because you kind of have to ask, is the image noise dominated in the sense that it's the limiting factor when you run clean, does it eventually just start cleaning noise? Or is it artifact dominated? At some point, are you starting to find that the brightest thing is actually an image artifact from bad calibration or bad data in some way? Is you can get, image artifacts, and if they're the brightest thing, it'll start saying, oh, this is a real source, and I'll put a clean component on it. And that can actually make things worse. And so you want to prevent that. And so knowing which of these two applies to your data, you can't actually know until you try generally. And it affects how you want to set clean thresholds. Because if you're noise dominated, then you don't really need to worry about artifacts causing problems. And you can just clean down as deeply as you want until you get close to the noise level. I mean, you don't, up, down until basically you're starting to generate clean components on noise, which isn't useful. And there are some opinions as to how low it's safe to go. Some people say don't clean below 3 sigma. Some people say, yeah, whatever, clean to half a sigma if you want. I got into a long argument with a postdoc during my PhD about this, because he insisted, no, you can just clean down to arbitrarily low because noise clean components don't do any harm. And that's not strictly true, because noise clean components end up clean, they get restored by this Gaussian clean beam. And the Gaussian clean beam, as going back to what I said about the flux measuring problem, the Gaussian clean beam is positive definite, so it contributes a net positive flux for every clean component, whereas unclean flux always integrates out to 0. And so if you start adding noise clean components, you're adding net flux that you shouldn't have, and that can mess with your image statistics in unexpected ways. So yeah, it's kind of long story short, don't clean too deep into the noise, I guess. And then when you start to get artifact dominated, you kind of need to stop the algorithm before you're putting too many clean components onto your artifacts, because that'll start really messing with your image in some cases. And so if your artifacts are 5% of the peak flux, then you can only clean down to 5% of the brightest thing in your picture. And so that tends to be a problem. And so if that level is too high, then you need to kind of go back and improve your calibration to get rid of the image artifacts. So yeah, under and over cleaning is sort of terminology that comes up a bit in terms of, are you applying sort of too few clean components versus too many in terms of setting thresholds and iterations. And so under cleaning is when you've not done enough iterations, or you've set the threshold high enough that it stops too soon. And that means that you have flux that wasn't captured in clean components. And so the flux density or intensity is still sitting with side lobe structure present. And so you have all of the side lobe structure, which can sometimes mess up things like flux measurements, integrated flux measurements. And so it can make it hard to get certain types of measurements depending on what your science goal is. Over cleaning is when you tend to have too many clean components and they're not locking onto actual signal. They're just giving you artifacts or locking onto noise. And ideally, they don't cause any problems, but I think that sometimes they can. They can actually cause, in the worst case, you can actually get the noise level going up as you clean, because normally you expect as you clean, you're getting rid of the side lobes. The noise level actually drops as you clean, and that's usually a sign that it's working. If things go badly, you start cleaning on noise, you start cleaning on artifacts, you start adding all this sort of incorrect signal, and it can actually cause the noise to increase. And this is when that happens, they say clean is diverging. And most modern imagers are good at sort of capturing when this happens and stopping automatically. Is a lot of imagers will actually pay attention to the noise level, and as long as it's going down, they're like, okay, we'll keep going. And as soon as the noise starts going up again, they're like, all right, I'm going to cut this off, even if I haven't reached the user parameters yet. So sometimes the imager thinks it knows better than us, and sometimes it actually does, I guess. And so in terms of sort of adding additional complexity to the algorithm, one sort of additional way of running clean, I guess, is the use of clean boxes. And so these are sort of user-defined regions or boundaries in the image where we say, okay, clean can look for peaks inside these clean boxes, but not outside of it. And so this is good if you can put a clean box around a source, but that excludes like the artifacts that are around it. So then you know that you're only putting clean components on the actual source, but not on the spurious detections. Downside of this is that it requires human intervention normally, is you actually have to look at an image and say, okay, I recognize this is a real source and this is a real source, but that is an artifact, so don't put that clean box there. And so that you have to put this sort of extra effort into doing it, with the idea that it does actually make things better in the end. And so the interactive mode in CASA that I mentioned lets you do this, for example, is you can run some number of iterations and then stop and then say, okay, am I seeing fake sources outside of clean boxes that I think are trustworthy and real? And then you can kind of add that. Or you think, okay, this source has become a complete mess of artifacts. I'm going to remove a clean box so that it doesn't make it any worse. You can have that kind of control at the cost of investing time and effort into making it work. Initial kind of source of problems are sort of extended sources, sources that are resolved and you can sort of have structure. I mean, because the clean algorithm assumes everything is delta function-like. And so as soon as you have a source that isn't a delta function, it kind of breaks it a little bit. It works decently well on sources that are partially resolved, sometimes even well resolved, depending on the source. It performs better than you might naively expect. And so it's usually okay. But you can find cases where it does tend to give you very weird results. And I think I have a couple of pictures of how that can kind of look later on. But it's the kind of thing, it's like if you do have a very large source in your image, you kind of have to keep an eye on it to make sure clean isn't doing something weird. And that's a vague statement because something weird is not a scientific judgment. And so this is why radio imaging is such a pain in the ass. Yeah, so there's a number of variations on the basic clean algorithm. The most common one, and the one that is basically implemented everywhere these days, is called Clark clean. And it's essentially a more efficient variation on the original Hoogboom clean. And that's because the Hoogboom clean had this subtraction step where it was sort of taking the synthesized beam centered on the clean component on the source, and then subtracting the synthesized the convolved sort of side lobe, the beam, over the entire image to get rid of all the side lobes. And so you're doing a subtraction over your entire image on every iteration, and it's computationally expensive. And so the Clark algorithm cheats. And so it divides the whole clean process into sort of major and minor iterative cycles. The minor cycle is basically the same as a single iteration from before, except that instead of subtracting the synthesized beam across the entire image, it only does it in a small area where the side lobes of the beam are sort of really strong. And so you're saving the calculation way out at the edges of the image where your side lobes are close to zero. So the calculation is not really doing much. And then what it does, which is a little weird, but computationally very efficient, is it takes your clean component model and it Fourier transforms it into visibilities, into a visibility model. And so what that's basically saying is, okay, I have these clean components. Here is what they should be doing to your visibilities in terms of changing the signal. And so you can subtract that off from your visibilities. And what you're basically left with is visibility residuals, all the signal that's not in the model of the model image. And so you can then just re-image with your residuals and say, okay, here's all the flux that's not in the model yet. Do a bunch more clean components, create a new model, a better model. And so you can iterate through that many times, and it's computationally more efficient. And in some ways, it's actually better in terms of reducing artifacts because you're actually subtracting the source and the visibilities. And so certain types of image artifacts can be removed by that. So those will be artifacts associated with the gridding process or sort of related effects to that, I think, which are usually not that significant, but if you can get rid of them, why not? And so at the end, you sort of get your final clean component model, create a final visibility model, convolve that with your Gaussian to get you your sort of clean beam. And then you can sort of add on the residuals and the visibilities and re-image. And the end result of that is you get a cleaned image like a Higgs clean image, but you've saved sort of, along the way, you've saved a lot of computation. So you can do a lot more iterations a lot faster. And so that's what's usually implemented these days. And so if you see in CASA major cycle, minor cycle, stuff like that, that's sort of what it's doing in the background. Another variation that is very useful is multi-scale clean. And this is actually quite recent. The paper describing it came out in 2008. And so I can say I've been doing radio astronomy for longer than multi-scale clean has existed, for whatever that's worth. But the idea is that extended sources are not delta functions. And so modeling them as a collection of delta functions is not really a good model. And it leads to a lot of artifacts typically. And so instead what it does is it says, okay, let's consider a family of models that have some width parameter. So you can sort of say, you know, I'm looking for sources that have some shape, but they also have some width that can be adjusted. And so you end up with sort of a parameter space of clean component models that involve the position in the image and this width parameter. And then as a function of those three parameters, you can have the flux density of a particular clean component. And so you can basically explore that parameter space, identify the clean components for whatever the brightest part of that parameter space is, subtract them off as before, and eventually build up a model image that includes things that have this potential width. And based on the original paper, the standard model is some kind of truncated paraboloid thing. The actual shape isn't really that interesting, but it sort of lets you build up an image out of those models, essentially. The tricky bit with this is that you have to define a list of potential scales, potential widths, to be sort of tested, because it's not going to explore the entire parameter space of possible widths. That's way too computationally expensive. And so the user specifies some list of model widths that it wants to test and use to build up your model image. And so there's some balance between trying to get all the scales that actually correspond to the sizes, the actual sizes of things in your image, while trying not to waste computations on sizes that aren't really relevant. And so the net effect is that this becomes very good at picking out these kind of broad extended things. And so this is an example from the original 2008 paper, where on the left, that we can see sort of the restored image created by these clean algorithms between the sort of traditional Higgs clean and the multiscale. And on the right is the residual. So this is the flux not captured in the clean component model. And so you can see the Higgs clean leaves a lot of diffuse emission present. It's below the clean threshold, but there's a lot of signal there. And if you were to like basically integrate over the full thing, there's a lot of flux density is in this diffuse emission. And the multiscale clean is able to capture that. And so the final image in the multiscale is a bit smoother. It correctly puts in all of this extended emission. It's not trying to build a model out of little delta functions. And so when you have something that's well resolved like this, you get a much better result. So I guess one more variation quickly is there's the multi-synthesis imaging process has its own clean algorithm. I don't actually know anything about it. I don't even really understand how that works, because I don't understand how you can add together clean components in spectral index. That's not a meaningful operation, and so I don't know how they've solved that, but they apparently have. And so you can apply clean to multi-frequency synthesis to get really, really pretty pictures. And then sort of the ultimate imaging algorithm at the moment is multi-scale multi-frequency synthesis, where you combine the multi-scale clean with the multi-frequency synthesis, And so you can basically capture all the frequency behavior through the multi-frequency synthesis to get the the spectrum information out and all the resolution information. And you're also capturing all of the extended structure and all these highly resolved sources through the multiscale. And so this is sort of the ultimate imaging algorithm as implemented in CASA these days. If you have broadband data and data that has well-resolved structures in it. So any other questions on clean before I move on? Yeah, so this is sort of a lot of variations on sort of different aspects of clean that all kind of boil down to it's messy, it's complicated, and it takes a lot of iterations to get the best result out. Okay. So I wanted to talk a little bit about calibrating interferometers, because this is an important process if you're dealing with your own data, and it's not really clear what all the steps are or what they do. So I wanted to kind of cover that in terms of the sort of background structure of the problem. And so for this, we are going to go back to the Jones matrix formalism, where we're getting some signal out of the individual telescopes, VA, and that signal is related to the sky signal, but it's been modified by all of the properties of the electronics and the dish and whatever else. And so for every antenna A, we get some output signal that's been modified by this Jones matrix J. And this Jones matrix depends on a lot of different physical effects that we don't necessarily all have to care about. So the Jones matrix it's a 2x2 complex matrix, and so that gets you 4 complex numbers, giving you 8 parameters. And so in principle, then we need to solve for all 8 of these unknowns to understand what the actual transformation has been done to the data. And then we can potentially invert that to recover, take the measurements, invert it to get the actual sky signal so that we know what the actual signal was originally. Some of these factors are usually sort of treated separately from the calibration process. They can either be done analytically, or they're potentially either can't be solved in the available data or they're not important. And so stuff that can be solved for is usually split out and done mathematically directly in the algorithms. And anything that can't be solved for just gets lumped in, and you get these 8 unknowns. There's a couple of factors that don't appear in the sort of original Hamaker formulation of this. And so there's a section actually in the CASA cookbook that goes into this in somewhat more gory detail if you're interested in. But the principle of it is basically the same. And so, yeah, so here's kind of the sketch of the process. Is you have some sky signal coming in, and we split this into sort of the X and Y polarizations. Get collected by the antennas, goes through some electronics, and then you get your voltage signals get fit into the correlator. So just to avoid confusion, the P and the Q here that Hamaker uses refer to the 2 orthogonal polarizations of the antenna, and he left it general so they can either be like an X and Y dipole antennas, or they can be the left and right circular antennas. And it's the formalism is general for both. It just changes some of the matrices involved, and he left it general. But so in general, yeah, this is like your X and your Y dipoles in each of the antennas. And so the mathematical structure, which I appreciate not everyone will be interested in. I rather like this because I think it's beautiful, but as sort of background. So Haslam ends up defining this visibility vector. These are all the products, which are the correlations essentially. The correlations of the 2 signals can basically be expressed as a product of Jones vector elements. in the mathematical formalism. And so the process of measuring a visibility then ends up being this outer product of the signals from the two telescopes. And the signals from the two telescopes are modified by these Jones matrices. And so you end up with this outer product of Jones matrices and then the actual correlation of the sky signal. So this would be considered effectively the true sky visibilities. And then this is a complicated matrix that describes how the visibilities are modified from the actual values to the measured values. And then the idea then for calibration is we want to solve the unknowns in the individual Jones matrix for every antenna in order to, basically we're trying to solve for all the unknowns in those Jones matrices on an antenna by antenna basis. Because most of the effects that we need to calibrate for are antenna dependent effects. So the electronics are different for every antenna. The antennas are at different positions, things like that. In theory, you can have baseline dependent effects where something that affects only a single pairing of two antennas, whereas those two antennas matching to other baselines, other antennas aren't affected. But that's not very common. I don't actually know what the physical origin of baseline effects are. And most calibration schemes these days don't actually have any calibration for those because they don't tend to be very large. And baseline effects tend to have, if you try to calibrate for it, you end up with way too many free parameters. And so there's a lot of risk for overfitting your data sort of, or over calibrating it. Maybe you're having your calibration not being constrained, but basically everything that normally affects the calibration of an interferometer is always antenna dependent. And so we can go through this Jones formalism where everything depends on the Jones matrix for the antennas and not on any baseline dependent stuff. And so the calibration process then requires some observations in which we know what the sky signal actually is. And so we can then take the sky signal that we know, the measured visibilities, and say, why are these different? And what Jones matrices do I need to make them the same, effectively? And so if you kind of expand it out a little bit, starting with this, the Jones matrix equation from before, expand it out in terms of the matrices, these are the parameters that we have to work with and the ones that we're trying to solve for. And so these can be sort of lumped into different categories. There are the gains, which sort of the gains or the amplitude calibration parameters. And so these are effectively defining the conversion from whatever units your measurement is in to sky intensity in Jansky's per beam or equivalent, or that conversion or vice versa. And so that's important for getting sort of the units and the amplitudes and the flux densities all correct. And so the gains are generally a function of frequency. They're affected by things like your bandpass filters, your amplifier response. And so you need to correct for all of that in order to make sure you're getting the correct flux density as a function of frequency. They tend to be a function of time because the electronics tend to fluctuate around based on changes in temperature or other environmental conditions, such like that. And they're a function of antenna. You have to solve it for every antenna because they have independent signal chains. And sort of the other key parameters are the phase parameters. And so these represent the signal, the change in the phase introduced by the system. And if you don't calibrate for these, you end up with the wrong phase in your visibilities. And so the phase, as pointed out last week, basically encodes position information on the sky. So if you get the phases wrong, you end up with positions being wrong on the sky. And so these phases are a function of frequency and time for the same reasons as the gains. And they can actually also be a function of direction on the sky. And so this is because not all of the phase effects are actually in the electronics. There's also phase effects caused by the ionosphere or the atmosphere. And these can then change depending on, as you look in different directions in the sky, you're looking at different parts of the atmosphere, different parts of the ionosphere, as well as you can have the different antennas, again, are actually at different locations, and they're looking through different parts of the ionosphere as well. And so here's kind of a sketch of why this is a problem. Is if we start off with a plane wave coming in from space, it is, in the ideal case, it is a perfect plane wave. And then it hits the ionosphere and the atmosphere, which acts as a phase-changing screen, because at low frequencies, the ionosphere, the plasma in it, has a index of refraction that can changes with density, and so it can change with position in the ionosphere as there's density fluctuations. And so the index of refraction changes, and so that changes the shape of the wavefront and the phase behavior. And at high frequencies, the same thing can happen in the atmosphere, as you get some index of refraction effects at very high frequency radio emission. And so the same effect is what comes in as a plane wave becomes slightly distorted because of all of these atmospheric effects. And so you need to calibrate those away to basically get the equivalent of being able to treat your incoming signals as perfect plane waves, because that's what all of the interferometer derivation assumed. And so we need to basically correct for those effects. And so the final terms, for the off-diagonal terms, are the polarization leakage parameters. They're also called the D terms in Haslam, because he factored these out as a separate array, or a separate matrix called D. And what these do effectively is, this is what causes signals of one particular polarization to be observed as a contribution in the opposite polarization. And so these are effectively sort of false signals in sort of the polarization response. And this can be caused by things like imperfections in the antennas. You can imagine having cross dipoles, but maybe they're not 90 degrees, maybe they're only 88 degrees. And so there's some mutual sort of mutual signal being detected by those. And so you can have those kinds of flaws occurring. And they're a function of antenna usually, because each antenna can have slightly different sort of physical construction. I don't actually know if they have time or frequency dependence. I've not dug deeply enough into leakage terms to know what kind of time or frequency effects there might be. Frequency effects probably, because you can imagine certain flaws, like maybe the shape of the dish is flawed, and then that scales with wavelength in terms of optical effects. But time, I don't know actually. So polarization leakage calibration actually only really matters for people who are interested in the polarized signals. For unpolarized, or if you don't care about the polarization information, you can get away with just doing the gains and the phases of the two orthogonal polarizations. And so I wanted to put in a quick word on sort of the number of free parameters here, because this is sort of a mildly interesting aspect of things. So for sort of a normal continuum calibration for an interferometer, so not worrying about the polarization linkage, you have 2 times the number of antennas gain parameters, because there's an X and a Y. So you get 2 per antenna. And you get 2 times the number of antennas minus 2 phase variables. And so the reason for the -2 is because we have no absolute phase reference. So when we measure the visibilities are always recording a difference in phase between the two antennas in the baseline. And so the visibilities always reflect differences. And without a phase reference, there's no way of actually recovering absolute phase, although absolute phase isn't interesting. There's no physical information there. All the physical information is in the differences. And so what's normally done is the user or the software selects a reference antenna, which can just be sort of any of them. And it says, okay, I'm going to use this antenna as my phase reference. I'm going to set its phase to 0, and all the differences are going to be measured relative to that. And so you lose 2 free parameters. So one antenna phase reference is X and Y. So you have you lose those 2 phases as just being set to 0 because we have no absolute phase reference. Although if you are doing some polarization calibration, if you want to do polarization measurements, you actually do need to reference one of those phases, because in these equations, X and Y are decoupled from each other, aside from the leakage terms, which are sort of a flaw term. And so the X and Y phase measurements and the X and Y sort of output signals are completely decoupled from each other electronically. But this is a problem if you want to do polarization, because the relative phase between X and Y contains information on the polarization state of the wave. And so you don't need that for total intensity, but you do need it to get the polarization state. And so you have to measure what the relative phase is between X and Y. And so that requires an extra calibration step, which only has 1 free parameter. So it's fairly straightforward if you have the data for it. If you do polarization leakage calibration, you end up with 4 parameters per antenna, and that comes out of these having both an amplitude and a phase contribution, since they're both complex numbers. So you end up with four parameters to completely solve for that per antenna. And so the reason this is kind of significant is to contrast it with the actual number of measurements that we have to solve for these parameters. And the number of measurements is the number of baselines, which is the number of antennas, well, with the number of antennas it can baseline with, so n minus 1 over 2, times the 2 different polarizations. And so this number scales it scales basically as n squared, number of antennas squared, whereas all of the calibration scale is the number of antennas. And so you very quickly have many more measurements than you have unknowns, and so it's a very well-constrained problem, as long as your number of antennas is more than 5 to 10. And so this becomes very good because solving for all of these unknowns for the calibration process is sort of better constrained, so the errors in the calibration get smaller effectively. You have better signal to noise in your solutions. And so this is why the larger the array, the sort of better the calibration quality tends to be automatically. Any questions on sort of the mathematical framework before I get into sort of the practical application? Okay, I'll assume you're completely lost, perhaps. But sort of on the practical side. So the calibration procedure requires a calibrator observation. The ideal calibrator is one that is bright, isolated, unchanging, and unresolved. And the reasons for this is you want a bright source because you want to have the best signal to noise in your visibilities to calibrate on. You want an isolated source because you don't want to have other sources affecting the actual signal of your visibilities, because ideally this whole process is going to involve having a model of your calibrator and comparing the data to the model. If there's other sources, you either have to include them in your model, which makes the model more complicated, or you ignore them, and suddenly that's going to affect how well you can calibrate. Your calibrator needs to be unchanging. It has to have no time evolution, because you need to have a model, and you need to be able to say, this is what my source is like at the time I observed. And so if your source is evolving over time, you may not actually be able to say, I know exactly how bright this source is today. And so this is why monitoring campaigns for calibrators is a big deal, because people at the VLA want to be able to say, okay, this is our calibrator, and we know it hasn't changed brightness in the last 20 years, because otherwise it's hard to know that your fluxes are accurate or your flux densities.
What about the polarization?
Yeah. So what I'm going to discuss is mostly independent of it's mostly not considering polarization calibration. So you can calibrate for gains and phases without knowing anything about the polarization of the calibrator. In order to calibrate for the XY phase difference, you need to know the calibration, or you need to know the polarization. And you need to also know that it hasn't changed. And in order to calibrate for the leakage, there's two ways of calibrating for the leakage, depending on what kind of telescope you have. And I didn't write slides about this, and I kind of ran out of time to think about it. But to do a leakage calibration, you either need a source that has no polarized signal at all. So you can say, if I detect polarization, it has to be leakage, and then you can model the leakage that way. Or you have to have a source with a very well-known polarization. So you can say the deviation from what is known is caused by leakage. Yep. And so there's a couple of different ways of sort of handling that, in terms of, some people say observe both an unpolarized source and a polarized source separately. Some people say you don't have to. That kind of gets into its own kettle of fish in terms of opening up new problems. Yeah. Yeah, and so also the reason an unresolved source is good for calibration is because an unresolved source, one that's close to being a delta function, is, sorry, distracted by chat. The reason an unresolved source is better is an unresolved source, or one that's effectively a delta function in the image, Fourier transforms into having equal brightness across all visibilities, across the entire UV plane. And so if the brightness is the same everywhere in the UV plane, that means you have equal signal to noise for all baselines, even for the very long baselines. If you're, sorry.
Is this calibration procedure for the interferometry or for single station?
Yeah, this is for an interferometer.
Okay.
So single dish calibration, I don't know enough on to comment, so I deliberately avoid the topic.
Okay.
Yeah, sorry. I thought about it, and it's like, I don't have time right now to get up to speed on single dish calibration. So my apologies for that. Yeah, so some of these parameters, like particularly being unresolved, aren't so critical if you have a good model for the calibrator. So an unresolved source is easy to model. It's a delta function. It has like three parameters, and that being the X and Y coordinates and the brightness. So that's the easiest thing to model. But you can model with more complicated sources, and for example, the VLA has a set of standard calibrators, and they actually found that some of their calibrators become resolved at very high frequency. And so they had to generate a whole set of very, very good models for their calibrators to include that kind of structure that starts to resolve in at high frequencies with long baselines. And so the process then is, yeah, you take your observation of your source. Particularly, so typically you start with a gain calibrator, which is very, very bright. Compare the measurement to the model, and you can use that to determine the gain values on a per antenna basis, per polarization, so separately for the X and Y. And usually there's some sort of time chunk and frequency chunk size, which is a user parameter. And the reason that's done is you try to integrate over some number of measurements, either in time or in frequency, in order to maximize your signal to noise. Because if you try to calibrate on every single measurement, which might be every like 1 second, you might not have enough signal to actually get a reliable solution out. On the other hand, if your solution interval is really long or really large in frequency, you might accidentally integrate over structure in the Jones parameters, and you would lose some of that structure and you wouldn't be able to properly correct for it. And so there's a balancing act between having good signal to noise to get reliable solutions and having the smallest possible sort of intervals for your solutions to make them sort of to capture the most variation in time and frequency. And there's no easy recipe for this either. The next step after that is to observe a phase calibrator, which is close to the target on the sky. So this is to address the direction dependence of phase effects, to the phase effects from the atmosphere and the ionospheres. You observe in a direction as close to your target as reasonably possible, and then hope that the phase doesn't change that much between the phase calibrator and the target. And so this is often necessary because there's only a small number of good gain calibrators, usually like 3 or 4 in the entire sky. So you can only pick like 1 and you can't control where it is. So you observe some gain calibrator in wherever it happens to be, and then you pick from a much larger number of phase calibrator targets, the one that happens to be closest to your actual target. And so you go through the same process of trying to optimize the phase values by finding the best solutions to the Jones phase parameters. And so once you've gone through that part of the process, you can observe the target and take the gain and phase solutions that you found from your calibrators and apply those to your target data. And so then you have target data that has been correctly calibrated for gain and phase in principle. Although, because there is time variability potentially involved, you generally repeat the phase and the gain calibration every so often during a long observation to make sure that if there is time variability, you can kind of interpolate between to capture that in the target data. And the time interval for that depends a lot on the time scale of variability in the system, and particularly phases. So phase stability changes a lot with frequency because it depends how strongly you're affected by atmospheric or ionospheric changes. And so at very high frequency, where the atmosphere starts to mess with your phases, you may only be able to work for 10, 15 minutes before the phases start to change enough that you want to recalibrate. And so you start to, for those kinds of observations, you'll observe your target or start with observing the phase calibrator, get your phase solutions, observe the target for 15 minutes, do another phase calibrator measurement, and then you can interpolate the phase, how it changes between those 15 minutes and apply that to your data. And so that sort of repetition of the process over the length of an observation is necessary for that. And then finally, in some data, there is the option to self-calibrate, which is the next topic. So are there any questions on that up to that point? Okay, so self-calibration is an interesting process, and it basically involves calibrating, as the name suggests, it's basically calibrating your data on itself, like on your target data. And most people's reaction to this is, how can you even do this? Because your target data is the measurement, the scientific measurement you're trying to make. How can you calibrate on something if you don't already know what it is? And that is a very good point. And the reason it works is twofold. First, it's usually just for the phase errors that get introduced by the shift in position from the phase calibrator. And the other reason it works is because if your signal, as long as your signal to noise is good, the difference between the number of free parameters and the number of measurements you have means that you can't really mess up the data too much. The data is sort of naturally resistant to being calibrated into sort of the wrong shape or configuration. And so the process of doing this is you start off with some initial calibration, which is what your gain and phase calibration is for. I mean, the gain is also important to get the flux scale correct. But so you have your initial calibration, you image it, and then you can make a model of the image, which can either be, you can take the clean components is the easiest way of doing it, or you can also use source finder output. If you have a source finder that characterizes your sky in terms of sources with some well-defined model geometry, is you can build up this model and then say, basically apply a calibration procedure. You're saying, okay, I have a model. How does the visibilities compare to the model? Can the phases be adjusted to make the data more like the model? And so you can do this, and then you can repeat this process, make a new image, see if the image is better, which it usually is, and just repeat this until the image gets as good as it gets, or until the algorithm is saying, no, I'm not changing the phases anymore. Because usually after the first image, you can generate a slightly better model. And this works mostly on phase, because the phase encodes position information. And so if the phase errors are significant, it tends to blur out the source a little bit. But the location of the source is usually okay. And so that can be used to sort of generate a model. And so as you do this and you go through the self-calibration, it tends to sort of sharpen up a source as it gets the position more correctly, and it fixes the phase errors that are blurring it out effectively. And so an example of how this can look, this is one that I took from a LOFAR paper, and we're seeing 4 different sources down the 4 rows, and stepping from left to right is iterations in a self-calibration process. And the numbers on each image are the noise, the local RMS in the image. And so you can immediately see how the initial image is pretty rough. There's a lot of artifacts. This is after clean. So these are image artifacts left after clean. And in some cases, there may actually be clean components on the artifacts because they're bright in some cases. And then so after the self-calibration, it starts to firm up the images, put the flux where it should be, and that reduces the artifacts because the flux is going to where it should be and not being scattered into different locations. And so after a couple of self-iteration cycles, everyone gets a little bit better, and you're sort of better able to model and image the data. And so the improvement in the noise is quite remarkable. It can be up to a factor of 10 if you have really bad data. And so this is a really useful algorithm if you have data that is strongly affected by phase errors like this. And so whether that's true depends a lot on what frequency you're working at, on what scale, like what resolution, stuff like that. So it's not always necessary, but it does help some data sets by a lot. Okay, so I'm kind of going through a lot of this process in reverse. I talked about imaging, then I talked about calibration, and now, before that, I'll ask for any last questions on calibration before I move on. Okay, so yeah.
Sorry.
Oh, yeah, go ahead.
What if you have a nearby bright source that eventually showed on the image? So how do you make this selection preference so that the cleaning does not target the wrong source?
Well, I mean, if they're both, if you have more than one real source, then it's fine. You probably want clean components on the fainter source as well. But if you have artifacts like in this image, second panel here, you can see there's a bright artifact over here. If you want to prevent that from being cleaned, you need to go to clean boxes effectively, which means doing it manually.
Oh, okay, I see.
Yeah, so doing it automatically will probably put clean components on it. And one of the nice things about so in the context, for example, of self-calibration, is even if you have some clean components in the wrong spot that, from artifacts like this, even if that's in the model, because there are so many more measurements than there are free parameters, it's possible, it's generally safe and it doesn't mess up the self-calibration too much. And so after a couple of rounds of self-calibration, it'll sort of naturally fix itself normally.
Okay, cool.
But that has actually reminded me of a funny anecdote about self-calibration. So a friend of mine during my PhD did a school on VLBI, on very long baseline interferometry, which includes self-calibration. And as a sort of experiment to himself, he played with sort of a pathological case where he ignored the actual sources and put clean boxes on noise in the shape of a smiley face. He did this sort of little smiley face with eyes. And he used that as his clean boxes and generated a model of that and self-calibrated to that model. And sure enough, after a couple of iterations, he ended up with an image of the sky that looked like a smiley face.
Amazing.
And a little frightening. But I discussed this with him, and I think we kind of agreed that the reason it worked is because the VLBI data has a small number of antennas. And so the amount of redundancy that I was talking about goes down by a lot. If you only have 9 antennas, say, you get 9 times 8 divided by 2 is 45 baselines. Am I doing that right? Whatever, some number of baselines like that, 40-ish baselines. And the number of parameters is 10 or 20. And so it's not a very over-constrained problem. And so that's how you can get those kinds of problems, I think. So he had to work hard to make it work, even with that. So I'm not particularly afraid of self-calibration because of that. But yes, if you try hard enough, you can make a smiley face on the sky with radio interferometry. I don't recommend it, but if you have more time, then you know what to do with.
So self-calibration isn't meant to be something that replaces the generic clean algorithm, but something
No, this is on top of clean.
Right. Okay. I just wanted to make sure. Okay. Awesome.
Yeah. This is sort of catching artifacts that clean stumbles on. Is because you have phase errors, you can end up with little lumps of emission like these, and those will produce false clean components. And so by improving the calibration, you get rid of those, and suddenly clean does better, because clean assumes everything is shaped like the synthesized beam, and these artifacts are not shaped like the synthesized beam. And so self-calibration makes clean better.
Is there an order for this? A preferred order for this calibration?
Not entirely sure what you mean by that.
Oh, like for example, which type of let's say the clean algorithm, then this type of calibration, or the other way around?
Yeah. So the clean algorithm is run every time you image, generally. And so every time you create an image, you do the imaging with the inverse Fourier transform. Then you run clean to improve the image, And then if you're doing self-calibration, you then take the cleaned image, which is the best quality image you can make, and you use that to generate your model. And in some versions of self-calibration, it actually takes the clean component map that is generated by the clean algorithm. Because the clean algorithm, it generates the restored image, but it also usually saves the clean component map, the image of just all the delta functions. And so it, lost my train of thought, sorry. So yeah, it also saves the clean components. And so those are used as the model sometimes for the self-calibration. So you run the imaging, you run the clean, you get your model from the clean algorithm, you feed that back into the calibration, then you make a whole new image is sort of the procedure for that. And so it gets computationally expensive because you're doing a calibration step, which is not that expensive. You're doing an image step, which is so expensive, and you're doing clean, which is rather expensive in terms of computational time. And so, yeah, like for example, on LOFAR data like this, doing an entire self-cal loop could take a week on a large computer, on a supercomputer. And so it can be a painful process on large data sets.
Sorry, so you said self-calibration can take up to a week, so it's a lot more computationally intensive than the regular clean algorithm?
Well, it's because you have to run clean on every iteration of it.
Oh, so okay, so like in total, like the whole thing.
Yeah.
Not so it's not just the self-calibration that would take a week.
No, the actual like the calibration part of it, like the process of actually solving for the phases and stuff, is fairly fast depending on the software. I think it's fast. But the actual, the fact that you have to make a new image every time means that every iteration becomes a bit expensive to do sometimes. And so you don't always necessarily want to keep self-calling until you've gotten the best possible image. You may just want to go until you have a good enough image, especially when you're on a deadline. Wow, I've actually managed to fill much more of this lecture than I actually expected on this. Okay, this is good. Yeah, okay. So I'll zoom through maybe the next bit, which is, I wanted to talk briefly about data flagging. 'Cause as I was putting this together, I realized I forgot this, which is sort of the first step people usually have to do with their data. So flagging is the process of removing anything any bad measurements, effectively, any bad data that's been incorporated into the data set. Because if you have bad data, it can really screw up calibration, and it can also screw up images. Like particularly if you have something like RFI, if it's 1,000,000 times brighter than the actual measurements, then when you're doing the calibration, you're finding the solutions. It's usually some kind of a least squared optimizer to get the solutions. If you feed something that's 1,000,000 times too bright into a least squares algorithm, it's going to go crazy and you'll get bad results out. And the same thing with the imaging process. And so there's a lot of sort of different possible causes. So yeah, man-made interference is a big one. Sometimes antennas can just be weird and they can glitch out or turn off or do different things. There can be data lost because of this. Something shuts off for a minute or something. The sun can be a problem, although you usually can predict that ahead of time where it's going to be and try to avoid it. So you need to try and get rid of this, basically. Get rid of this data so that it's not causing problems. And so the easiest way to identify it is usually if the amplitudes are really, really off, either too bright or too faint. And particularly if they stand out somehow in terms of time or frequency, or if one antenna is very different from the rest. Stuff like that. And so there's a lot of automated algorithms now that have gotten pretty good at picking out sort of the easy stuff. But sometimes, particularly traditionally, you needed to sort of go in by hand and figure out what was bad and manually sort of tell the code to get rid of it. And traditionally, quite often you would need, or even now, you need 2 rounds of flagging. So you do 1 round before you do any calibration to identify all the problems that are sort of immediately visible. But sometimes you'll have data that looks okay, and then after you calibrate it, suddenly you can tell, no, these aren't good numbers. Something is off about it. And so typically you'll run the automatic flagging twice to try and capture these problems. Although that can be a little bit dangerous in a few cases, because what can sometimes happen is you have some data that's reasonable, that's okay, but you end up applying bad calibration solutions. If something goes wrong in determining what the calibration should be, and you get wrong values, and then you apply that to the data, suddenly the data is now wrong and can look weird and get flagged out. And so sometimes it can be bad calibration, which can be fixed, and sometimes it's legitimate bad data. And telling the difference kind of sometimes needs a human being. So yeah, let's look at a couple of examples. I pulled some images from early on in my PhD with some LOFAR data. So here's what they call a waterfall plot, or this is all a variation of it, where we're seeing sort of frequency versus time, showing the typical brightness of the visibilities. And you can immediately see areas that stand out. So there's bright spikes. There's certain bands where various man-made signals have turned on and off, stuff like that. And so I ran this through an automated algorithm, and it picked out the worst of it. So like that bright spike is gone, all these little horizontal features are gone. The color scale has changed, so it's a little hard to sort of match them, but a lot of this sort of little, whatever this intermittent man-made source is, that got just destroyed. So the automated algorithm can quite often do a reasonably good job. Sometimes it doesn't. This is a fail. There was an entire group of bad channels here, and it didn't always get all the data flagged out. I don't even know what this is, but it's bad probably, and it didn't catch it. So this is why you sometimes need to go in with a human eye. And there's a lot of sort of different ways of visualizing the data in order to try to look at it. So we might get into some of that next week. But like here's an example. This is a scan just looking at the visibility amplitude over time, and there's like a minute or so where things were just weird. And when I sort of tracked this down, it was just one antenna that turned out to be producing numbers that were way too high for a minute. So something had gone wrong, but it had resolved itself a minute later. So the rest of the data was fine for that antenna. Yeah, same kind of thing. And this time I colorized it by antenna. So you can actually see there's a bunch of antennas that are weird at different times, different antennas. So this was a really messy data set in some ways to calibrate. Then after flagging, I got rid of the worst of it. There's still some left. And so this kind of shows that a lot of flagging tends to be a little bit judgment driven, because you kind of have to ask yourself, how much of a problem is this? Is it making things a lot worse? And how much time and effort is it worth to actually root out a lot of these little issues? So a lot of this stuff is kind of bad, but I don't remember if I actually went and fixed it, because at some point, sitting for a month trying to flag all these data points and identify little glitches will drive you neurotic. True story. But yeah, you can see it. So this is an example of something that didn't show up until after calibration. This data, this data sort of looked okay, and then I calibrated it, and something was clearly bad after that. And so I think again, this was like a bad antenna for a couple of minutes, and so I just had to flag that out manually. And so there's a bunch of tasks in CASA that let you identify sort of blocks of data, this antenna in this time range, in this frequency range, and sort of adjust those. And so it's something that is a little painful. It's very kind of human judgment oriented, but if you're trying to maximize the quality of your data, it can be really good. And so not all data sets will need a lot of attention, but traditionally this was the thing that people did to try to get the most out of what they had. So I'm gonna probably stop there. I didn't get to the radiation stuff, but that means I have slides ready for next week already, I guess, or for next time, next lecture. I had one image that I pulled since I was going through my old research notes. I have a long collection of plots and images that are basically failures. I pulled this one out because it's quite entertainingly bad. I don't know what went wrong. No, that's actually not true. According to the notes that I had, this was a glitch in the multi-scale clean algorithm in the LOFAR imager at the time. And so this is the same image that had that big galaxy in the center. And because I tried to clean it with multi-scale and the multi-scale was broken, I discovered the first galaxy with negative flux, I guess. Horrifying things happened. And so adventures in calibration.
I love failed plots.
I have a lot of these. It's entertaining to go. It's entertaining now to go back to.
You should post them on the Slack channel.
Tempting. Yeah, poke me about it when this is over and I have time. So yeah, radio emission mechanisms I'm going to save for next time. I did want to say, so what I wanted to do now is now that we've sort of gone through interferometry theory and a little bit of practical stuff, is I wanted to maybe do the CASA tutorial that I had planned next week. And so what I was kind of envisioning is I'll sort of lead off with some live demo stuff, sort of the key aspects of how CASA works and how it's structured, and sort of how you sort of typically do the main tasks. And then I was originally going to design my own sort of semi-tutorial thing, but I don't honestly have time. So there's a lot of probably very good tutorials on the CASA webpage. And so what I propose to do is people can pick one of them. I'll look through them probably tomorrow or Monday and suggest a couple of the ones I think look most interesting. And then next week, everyone can sort of go through them. I'd originally, when I put the course together, I'd envisioned this as all of us in a room and I can look over people's shoulders and comment on sort of things as you experiment with CASA. And that'll be a little different with Zoom, but we can screen share and sort of look at each other's interesting plots and discuss what they mean and help each other debug maybe. And so I propose that we spend sort of 2 hours doing that next week. And so anyone who's interested in that, please install CASA ahead of time. And
I thought you mentioned that we're doing emission mechanisms next week, or is that going to be?
Oh, I misspoke. That'll be the last lecture then. That'll get shifted over.
Okay, cool.
I misspoke. Next lecture, which will be 2 weeks. So yeah. So that's what I propose to we do next week. And so anyone who's interested in that, we can just hang out for a couple of hours and play with CASA. And I can offer whatever. And I know other people have done a bunch of CASA stuff before. And so those people are also welcome to hang out and offer their own experience. Because I think collectively we probably know a lot. And some people know things that I don't for sure. Because as a guilty admission, I have never reduced data in CASA. I reduced data in APES, which is the precursor to CASA from the bad old days. And I've reduced LOFAR data, which has its own software.
APES is so old.
It's scary.
Oh, LOFAR has its own software?
Yeah.
Ah, cool.
Bits and pieces of it use CASA, like the plots I showed. Some of those were from CASA.
Is that common for other telescopes also to have their own software?
Yeah. Oh, now you've triggered a pet peeve of mine again.
Oh no.
So it's gotten better, but traditionally, every interferometer had its own software. So the VLA used APES. ATCA had Myriad. The DRAO synthesis telescope had something called Matter, which I've never seen or used. And I think Westerbork, I don't even know what they used, or any of the other interferometers. So everyone had their own software. It all had different syntax, different bugs, different cookbooks, etc. And CASA was the proposal to try to standardize it And NRIO invested a lot of money in that, and they half succeeded in the sense that ALMA now uses CASA, because they also paid a lot for it. VLA uses it, and other packages or other telescopes like LOFAR and ASCAP use bits and pieces of CASA, because CASA doesn't do everything that people need it to do. And so we haven't completely solved the problem. So there's bits and pieces like CASA Core is some of the core tools, and that tends to be used by everyone, but other bits are kind of different telescopes. It's different how well they go along with CASA. So that's a pain. But one last comment since we're going over time, which is that for the very last lecture, we basically lost the most of the application stuff as well as the emission mechanisms. So I'll put the emission mechanisms into the last lecture now. I want to do polarimetry because it's a topic that I'm very passionate about and that I've kind of pushed off to the end because some of it is relevant to calibration and such. So I want to talk about that a little bit. And then I was originally intending this whole section on like applications of radio astronomy, but I started thinking today, another thing that might be better is anyone who has a radio astronomy topic that they are confused over, either something that I've discussed that's still confusing or something that I've not discussed, but that you've tried to learn in the past and struggle with, shoot me a message about that. And I can potentially, if I know it, I can try and put together some slides on it. And then we can try and tailor things to what people have found confusing to some degree.
I want to, first of all, I want to say thank you for putting this together. I want you to maybe, while you are selecting the tutorials we'll run on CASA for next week, maybe you can keep this in mind for some of all the theoretical work that we went through for a couple of weeks. Yeah, for someone like me, some of them have not made sense because I have not actually, worked with the data. I've not seen what they all mean, so it would be reasonable to look for the tutorial that would connect them, so that would make better sense. So it should just aid the understanding of what we have done before.
Yep. That is a really great point. So I'll do that.
Thank you.
And the other thing I might do is, because I think some of the tutorials, there may not be tutorials that cover certain aspects of it, but I do know there's CASA tasks that can sort of produce interesting plots that are relevant to some of what I discussed. And so in addition to the tutorials, I might write up a short document on, or do something on how you can use CASA to look at data that illustrates concepts. Some plots that you might not normally make during a data reduction, and that might not be in the tutorials, but that also are illustrative. So I can look at that. But yeah, thanks a lot for that suggestion. Yeah, I am over time. Any last thoughts? All right, then let's adjourn for today. Once again, thank you all for coming. And
Thanks, Cameron.
let me know
Thanks,
about
Cameron.
any thoughts, or as I said on the last slide, let me know about anything that you've sort of across all of radio astronomy that you really think is weird and confusing and you want to hear more about. And I'll do what I can for the last lecture. So,
Sounds good.
Yeah.
all righty.
Go back
Have a
and
good
think
night.
about what I don't know.
It's a hard thing. So,
Yeah. Thanks.
all right. Yeah. Have a good night.
Bye.